The Director of Development and Public Affairs here at the MDI Biolab. We're thrilled to have you here for our Science Cafe, and we are talking tonight about the extraordinary images and scientific potential of 3D microscopy. So some of you may realize that over the last 5 or 6 years, the MDI Biolab has really been investing in its microscopy resources. It's really been a huge part of the growth of our scientific programs, but also a wonderful way to educate and leverage science happening across the state of Maine, but also internationally. So it's allowed us to develop a number of international partnerships. And I want to just mention that MDIBL has currently, I think, 7 microscopes in our microscopy facility, which is run by Dr. Frederick Bonnet, who's here with us today as well. Tonight, we're going to be focusing on the newest addition to the family, and we want to tell you a little bit about how that came to be and some of this fascinating technology that has come to play in utilizing this microscope. So I want to introduce the folks that are with us here this evening. Dr. Fabian Voigt is actually with us as a visiting scientist for a few weeks this summer, and he is the inventor of this mesoSPIM microscope that we're going to talk about this evening. He's currently a postdoctoral fellow at Harvard. And before that, you were in Zurich, is that right? So again, one of these great international collaborations is a wonderful example. And next to him is Dr. Marco Pende, who is also a postdoctoral fellow here at the MDI Biological Lab. He's working with Dr. Prayag Murawala, who is next to him. So we've got the lineup here. These guys look pretty thrilled to be here this evening. But what I want to do is have a conversation with these guys, help all of you appreciate sort of where microscopy technology has, how far it has come. If you're like me and you remember being in high school biology class and using sort of a light microscope, we're a long way from that. And one of the things that we want to help you appreciate is how not only the incredible images that this technology is producing, but really what it's telling us in terms of a scientific story and how it's actually moving our research forward and our understanding of what's happening at the molecular level. So with that, I want to just kind of open it up. And Prayag, do you want to just run through a couple of these slides, or Marco, somebody?
Let Marco run.
Okay, all right, well, let Marco do it. So we noticed the costume coordination that we have here with Dr. Murawala. This picture was actually taken well over a year ago, but somehow he managed to pick out the exact shirt.
I don't
that he
have
had
any shirts.
No, I just wanted to recognize him again. So,
So tell us a little bit about how this microscope came to be and sort of what the process has been for you in putting it together.
should
You can start.
I can start. Okay. So we were there was a period of time where we were not sure what kind of light sheet we want to build, and then finally we decided for the mesoSPIM, and this was kind of we had more or less 1 month to decide for
Well, I mean, we were discussing for a long time. Marco has a very long experience working with light sheet microscopes and tissue clearing methodologies. And ever since Marco moved here, we wanted to have an equivalent of light sheet here at MDI Biolab. But then we were going back and forth with what we should have and source of funding. And at some point, the opportunity opened to write a proposal to an IGMS. And that's where basically we moved. And that would not have been possible without Fabian's support, because we have been talking to him For much longer time, we were discussing the possibility to have a mesoSPIM here at MDI Biolab.
Great. So Prayag, I want to just jump in. Tell us what mesoSPIM means. What is that acronym? What is that for?
Maybe I have to go. So the microscopy technique we're talking about here is something called light sheet microscopy, and that's a technique that is very, very unlike kind of the microscopes many of you are familiar with. First of all, it's not something that has an eyepiece, so you cannot put your, you look through it in a classical way. Instead we're having a camera, so it's very much like the smartphone camera, and this camera takes thousands and thousands of images to then reconstruct a sample in 3D, and it's called a light sheet microscope because we take a laser beam that's been shaped into a kind of a line focus, a sheet of light. So basically you take a beam and then you form it with some lenses and mirrors into that specific shape, and that allows us to kind of cut an optical slice through a sample. And so the way the microscope works is by taking this beam and basically we move the sample through it, take an image, and so on and so forth. I believe later on there's going to be an animation showing it. And the second thing, which is a bit unlike microscopes that you may be familiar with, is that usually you have like a microscope company, something that sells you this device, and you pay them, you send them a check, and then there's one box arriving. And instead, basically, the microscopes that I built, these are custom instruments. You can think of them as like prototypes, but in the case of the mesoSPIM, it's a bit in between. So it's a system where actually quite a number of them exist around the globe. I believe the current number is on the order of 20, 21. And mesoSPIM is an abbreviation for mesoscale, and then the SPIM thing is basically means light sheet microscopy, or the technical term that people have been using is selective plane illumination microscopy. So very fancy words to describe basically there is something that looks like a sheet of light, and the meso means we're looking at samples that are kind of in between the micro and the macro level, and that means like samples that are bigger than, let's say, a couple of microns, and smaller, and then smaller than, let's say, 10 centimeters. And so that means in the case of the mesoSPIM that what the systems are usually used for imaging samples that range from like a tiny little fish that's only like maybe a few millimeters in size, all the way to a whole mouse that's 10, 15 centimeters or so.
I mean, one thing for the audience maybe. So we have a scanning light sheet. So when you think about this is a point source, and when you move this point source very fast, you create kind of a line, and this way you create the light sheet, actually. And Fabian said, yes, it's not coming in one single box.
Yeah. So
this is what you see here. This is Prayag's office. So we were not sure where to put them, so it ended up just in his office. And one morning he came and it was full of boxes, and I said, "This is how it is."
So we decided to do this in the midst of COVID. So you can imagine what ordering all these different components. There were at least 1,000 different components for this microscope, and so trying to get them, we were all excited when Prayag sent a note and he said the first components have arrived, and they opened it and it was one single screw, and I was like, this is going to be a long process.
19 screws were missing in it. So I was like, something is wrong. Yeah, and the thing is, this is from different vendors, so it's not just one vendor. You have to, and sometimes the vendors are different here in comparison to Europe, and we were not allowed to order from outside of US, so we had to adapt.
Right.
Actually.
Right. So Fabian, you talked a little bit about open source and the fact that, there are commercially available microscopes, but you all decided to build your own, right? Tell us why. What is the sort of benefit? I've learned as the fundraiser, what that means is, there's always a new toy that we need. See, when you get one that's already packaged, it's like a done deal. No, now this is like a constant, we need to upgrade. We need to upgrade all the time.
So the story of how this specific microscope came about, basically, this is like what I did on my Ph.D. to develop kind of the first iteration, and then actually the system that others copied was iteration number 5. So there was quite a bit of development in between. And basically how this happened was that a colleague of mine who works in neuropathology in Zurich, who basically they're looking at like samples from diseased brains which have some form of disease, possibly Parkinson. What this lab is specifically interested in was actually prion diseases, mad cow's disease, that kind of stuff, because many still remember that kind of time in the 90s when this was very much on people's minds, and they're interested in kind of how this, these degenerative processes kind of spread throughout the brain, and so they wanted to look at lots and lots and lots of brains, and so they were trying a commercial microscope, and it didn't really work for them so well, and so then I came into the picture when I got approached and said, okay, can we fix this microscope somehow? And I tried fixing it, and I failed miserably. So it was this, and the company actually refused to send a sale, like an engineer out, and so I don't know why. It's like they were scared of a customer. I have no idea. And so at this stage, I'm the kind of person who's like, okay, let's build my own. And then there's a bit of a process because, I guess many of you, when you kind of like when you're looking for something, like a new camera to buy, you kind of know a little bit what you want, but are not quite sure. And then actually trying one might be the best way to figuring out what you really need. It's very similar with like scientists and their toys, right? They don't quite know what kind of toy they need until they actually see the real data coming of the thing. And then they know, okay, we can actually now use it to answer our questions. And it's very similar in that regard to everyday life. And so I started building these iterations of systems to figure it out. And then at some point, after they had their first microscope in the University Hospital in Zurich, other people approached me and said, oh, we also would like to have one. And then that was the moment I said, okay, how are we going to make this shareable? And of course, it would have been also a possibility to turn this into a company, but there's one issue, which is that there's a very, very complicated patent landscape in this around this microscope technique. So there's lots of players, lots of companies which hold a tiny little piece of that IP space, and so it's very difficult to operate. And that was one of the reasons why it's say, okay, let's make it open source, and also it's a much more streamlined process. And the other thing we realized, it's a in many cases, like also, for instance, the stuff that we're doing here right now during the upcoming weeks is actually you modify the system according to the specific needs of the specific components. And that's something, if you have a commercial system, it's like a black box. So ideally, there's nothing you can change about it. But for instance, when looking at samples, which are unlike a lot of microscopy samples, like the ones that Prayag and Marco are preparing, there's often no one size fits all solution from a commercial vendor. For instance, classical example is like the sample is far too large to fit into your commercial microscope. And so that's why it became open source. And the thing is, there's a whole movement nowadays in making science, like the hardware that is used to measure things, image things, open source. And the part of the reason is that if it's open source and you have all the software, you actually know what's going on, whereas it's a commercial black box, you may not know what's going on. There's actually not so great for reproducibility. Right? So there's this aspect to it, and the other aspect is also one of basically, usually such an open source system is far, far, far cheaper than a commercial system. And that means that it's more affordable. Now, in the case of these instruments, it doesn't mean necessarily it's cheap. It's just roughly a factor of 3 cheaper than the equivalent commercial system. Right? Still a sizable chunk of money.
It also builds kind of a community to have such an open source thing. So many people around the world work with the same microscope, and they develop bits and pieces, and they put it back into the
repository.
And this is how, for example, the software here was upgraded by Simon Watkins, I think, for fast recording. And so this is a complete different group than the original group developing it, actually. And yeah, Fabian mentioned updating according to your needs. So we have the skeleton of the mesoSPIM right now, but we were considering maybe going a bit higher resolution. So we worked with Fabian towards a solution for this. And as he mentioned, with a commercially available microscope, this wouldn't be possible.
So let's look at the mesoSPIM itself first.
Yeah.
Yeah, we have problems with this thing. Does it work?
Yeah.
Oh, okay.
So
So
this is,
I mean, we can maybe comment on this. So if you're well prepared, it's possible to build this thing in a day. And so here I'm doing, that's the lunch break.
That was the longest part of the day.
And here I'm helped by Evi Platonova, who then went on to build the next system. So that would be, this is number 3 that ever was built. So it was April 2018. And she built the number 4. And so at the end of the day, we're going to put a sample and you see this kind of blinking thing. That's when the data has been acquired. And if you want to check out the project, it's on this domain, mesoSPIM.org. So you can build it in a day, but I should say this, what takes a day as well is just getting all the stuff out of the boxes. Right?
Yeah. So regarding ordering, it was like 6 months of ordering,
Yeah.
fighting, and I mean sometimes the lead time of certain.
so many components actually arrived after we built the first version of
Yeah.
mesoSPIM. And
Yeah.
on his second visit, which is now, we are integrating some of those components as we speak today, basically. So this is what the mesoSPIM at MDIBL looks essentially. This is what Marco and Fabian built here. And the
Now it looks like a mess, by the way, because we disassembled.
Yeah, I mean, I guess for most of you couldn't even tell that this is a microscope, right? This is like, and this is because it's built from these kind of sets, it's like erector cell, like Lego for physicists, basically. and so you have cables. There's lots of cables, right? So and then these weird black things. These are beam blocks because you have some laser light leaking out, and you kind of want to avoid hitting your eyes with it. And so these things block the beam, and then there's lots and lots of components. The sample actually goes into that little space where the little red box is. Yeah, so that's where the sample. Yeah.
So
and exactly, sorry, yeah. And then this is what the actual sample looks like, and there's a mouse brain, so that's hanging upside down. So the left and right lobes of the brain are on the bottom, and then towards the rear is the in this case the cerebellum, the small brain.
And maybe you think why you can look inside, because the sample was prepared in a special way. So we use a technique called tissue clearing. So it's a way how we make initially not transparent sample transparent. The mouse brain is not transparent per se. And yeah, so what is shining here is a transgenic signal in neurons, green fluorescent protein signal in neurons, which shines when the laser light is hitting it, basically.
So a bit of a microscope, basically, if you want to talk, Fabian. So that's the laser box over there. Then
Let him show, actually.
Yeah.
I mean, yeah, I don't know if it's maybe super confusing. It's the top button?
yeah, the
Yeah,
top is
okay. So yeah, so this is a box which is basically, if you were to open it, it looks like a rainbow because you have 6 different lasers in there which cover the full visible spectrum from like very blue to very red. And so you have these individual colors and you use them to excite different dyes. So the dyes will glow in different colors, but because you want to look at multiple colors, you need different excitation lasers, and these are all combined in a couple of fibers which are here. And then the microscope is kind of symmetrical. It has 2 sides because we can create one of these light sheets from the left and from the right. And oops,
Yeah, this is how it works.
Yeah, now I'm already kind of. Can you go back? You can go back.
No, you killed it.
Yeah.
I'm sorry that I killed the microscope.
Surprise.
So the sample goes in here, then there's a light sheet from the left and right, and this kind of component up here is a set of stages, we call them. So that moves the sample around very precisely. And the bigger the sample, the bigger the stages have to be in a way. And that's actually one of the things that the commercial microscope, oftentimes you cannot exchange that part. There would be something where you would kind of, I don't know, take a chainsaw and cut half of the microscope off in order to fit the bigger stage in. And now because I broke everything, I'm just gonna have the responsibility back to you.
No, so this is how the light sheet is working, basically. So you have here
So-
transparent sample, here's a sheet of light. And this is how it looks like in real life. You have a kind of the light sheet scanning through it. And this is how the images are generated on the screen, actually. And from such kind of imaging stack, maybe I should go to the next one.
Yeah.
We create such type of images. So we create many planes, and then we combine these many planes into a 3D image. And this way we can reconstruct a mouse brain. So every dot which is shining right now, here is a neuron. Not every neuron is labeled in this brain, otherwise you would see nothing, just a glowing thing. And you basically can kind of fly through the brain this way.
Amazing.
Yeah, so Fabian did a very good job when he built it. Yeah, so
the important thing is also,
okay.
no, leave it there.
Okay, leave it there.
So the other important thing is during his second visit, Fabian and Marco are implementing another detection path, basically, that would allow us to increase the resolution of this microscope. So currently, the smallest pixel on this image is roughly 6 micron. And with this new detection path, it will go to 0.6 micron. So it's like a 10-fold more resolution of the same image. So it gives you much more detail. It's like you're seeing something from space, and whether you see a building or a person standing next to a building, that's the difference we are talking right now. So this kind of a higher resolution will allow us to see even the finest fibers, axons and dendrites in brain and many other tissues that we are interested in.
Okay, let's go to the next. Yeah, so this is, to put it a bit in perspective, what kind of projects we are doing right now with this type of microscope. So we are working on polycystic kidney disease. So you can see here normal kidney and here the kidney with cysts. And then we perform tissue clearing. So we make it transparent. And yeah, then we use the light sheet to create single planes. Here, these black areas are the cysts. And please play. 1 second. Can someone press the play button?
Roy, can you?
Or I can press the play button.
I think Roy may be trying to do, oh, no.
No,
I
do it. Sorry. Perfect. So what you see here is a scan through such a kidney. And this is the XY. 1 second. This button is on the weird spot. Usually it's not here. Okay. So what you see here is the XY plane, but to have kind of to quantify the cyst, you need from all spatial directions a very similar resolution. And so now we turn it and you see it. This is kind of the Z plane. It's when you see it from the side, basically, you can see that you have a very similar resolution. This way we can quantify actually the cysts in the kidney. This is done with the help of Frederick and the light sheet facility. Not the light sheets, microscopy facility here. So this is the process how we create kind of a mask from this cysts, and we further process it, we mask it, and then we load it to the Imari software, and this way we can kind of create a volume rendering of this cysts. And of course, please look. Yeah, here you see the kind of the natural image, and here is the overlap, basically, and this is the volume. And so what we can say about this, so we can say, okay, this is the volume of the tissue, this is the volume of the cysts in the tissue, and then we artificially defined some size. So we said, okay, we have 3 very large cysts, we have 107 mid-range cysts, and we have like 708 small cysts. Yes?
I think it's very important for the audience to realize I'm a medical doctor. I've been looking at polycystic kidney disease forever. And we are looking at the size and we're looking at kidney sulfation. And this is the first time that this has been done in the mouse. Really the first time. I mean, nobody, when Marco and has shown these pictures to medical doctors doing research in mice, and everybody immediately, could I please have these pictures?
Yeah. So one additional thing is that we just got either the left or right, I'm not sure. The left kidney, let's say. Flip the coin. So the left kidney and the right was kept with our collaborators at JAX, with Ron Crestagna. And what they do, they want to do an omics approach, so look for genetic markers, which maybe, so if I say this kidney had a lot of cysts and they were very severe, then they look in the contralateral kidney for specific genetic markers and maybe showing which are increased. So this is one of the projects.
So at this stage, another thing to keep in mind is that this microscope is not only available to the MDI-BL researchers, but also we have made it available to many other researchers in Maine, but also it is open to all the visiting scientists of the MDI-BL. So it provides a much wider service. And Frederick over there essentially takes care of providing this service to the whole Maine, INBRI, and many other institutions.
And so just last week, Frederick taught a course of students and scientists from Maine to utilize not only this scope, but all of the microscopes in our facility. And that's really a big part, or a big component of the mission of this center, is to, as I mentioned at the outset, really help people throughout the state of Maine access and utilize the expertise and the technology that's here at MDI-BL. And Frederick has done an amazing job of really just formalizing that process, making it accessible for people, and really sitting down with them and trying to understand exactly what question it is that they are trying to answer, and helping them figure out which scope, which technology is going to be the best and most efficient to answer those questions. So it's you know, it's fun to sort of see all of this, but as a scientist, it's also incredibly reassuring to know that you don't have to be an expert in every single one, right? That there's someone or a facility who can help you and figure out exactly which you know, technologies you need to answer those questions. So
in this case here, you see a different project. So this is embryology related. You see here amphibian embryos, which are tissue, so different stages of embryos, and they're tissue cleared. Here you have a dark field image, which basically indicates light scattering, and here you have a bright field image, more or less showing how transparent something is. And you can see we achieved tissue transparency in a couple of developmental stages, and we did antibody labeling, and then we used the mesoSPIM to image these embryos. And so this is another image where we injected dextran in 2-cell stage, and you see that the dextran remains on one side of the embryo. So we say it's not crossing the midline. And this is a process what we do afterwards. So here you have the embryo, then you tissue clear the embryo, we label it with certain markers. And then here, for example, which you see with the arrow, you see dividing nuclei. And here's the dye. And here you see kind of a quantification of cell sizes, which is a way to show that our method just works. So what you want to know is, like, if you look at these top cells at the animal pole, they have a different size than the large cells at the vegetal pole. And these tissue clearing techniques are usually kind of harsh, and sometimes you can have a deformation or damage. And this was a way kind of to show, so with the help of Frederick, we measured the volume of these top cells, and we measured the volume of the bottom cells. And we could show here that the size is kind of similar of the top cells and the bottom cells, which is a quality control for the method we are using. What else? Yeah. Now some beautiful images, which are more for imaging contests, which I didn't want this year. I don't know, I have no taste. So this is just a brain like the one showed before. This is a zebrafish. It looks kind of an X-ray. So I played a bit with wide field, and then I just overlapped it with fluorescent signal. What else do we have? These are skeleton shrimp from the pier. Just found them. So it's their end, but resulted in a nice image. And yeah, that's the end.
Yeah.
So
That's great. That's very cool. So, I mean, all of this technology, Fabian, is amazing. And we talked a little bit about certainly the helping understand sort of the biology of some of these processes that we're looking at. And we talked about the potential impact for human health and looking at things like polycystic kidney disease, right? Where just kind of, tell us a little bit about where you see this going. Like, how do you see, you're sort of just getting to use it here, getting familiar with the scope, but how do you see this moving your work forward? Prayag, can you give us some examples of things that you'd like to understand more about in terms of, regeneration and what this might allow you what questions it might allow you to answer?
Yeah, so one of the biggest strengths of mesoSPIM is to get a whole organism view that is normally not possible through the traditional method of sectioning. And particularly when I talk about the whole system view, we are talking about nervous system, we are talking about vasculature. These are some of the systems that is spanned throughout your body. And you want to understand how neurons are connected from one part of the body to the other part, how the blood vessels are connected from one part to the other, and how do they play a role in tissue development, tissue regeneration. How does it attribute an organism to do certain function that they do? So we have many projects running in the lab. So in one of the projects, we are trying to understand in mouse, how does mouse recognize and turn whether left or right? So a group in Denmark has identified a brain circuit in mouse brainstem. And this circuit essentially allows mouse to go left. So if you activate that circuit, mouse will go only left and left, basically. And then the question is, how does the brainstem neurons that they have identified connect with the rest of the body? So the spinal cord, and from spinal cord, they reach various parts of the body. So this is one example where we are studying connectome. The second example we already learned, that is the polycystic kidney disease. So what we are trying to do there is, as Marco was explaining, the left kidney. So we have created a huge diversity of mouse. This is in collaboration with Jackson Laboratory down the lane with Ron Costania's group. And what they have done is they have created a panel of mouse which are very diverse in their genetic makeup. And then they all have polycystic kidney disease. They have the same gene that causes the PKD. Now, what we are trying to understand there is how come some mouse have a severe defect or severe symptom of the PKD, and why does other genetic makeup does not allow high cyst formation? So the left kidney is basically used for this kind of tissue clearing and imaging and understanding the severity of the disease. And then the right kidney is taken, and we are doing the transcriptomics and omics approaches, which essentially allow you to identify genes and proteins that are differentially regulated between severe cases and milder cases. And that would allow us to identify the key driver of PKD, which then we can play around with. And we hope that if we can control that behavior, we can probably control the polycystic kidney disease even in humans. So these are just two examples that I described, but there are many, many more applications I know for sure that here, even at MDIBL, we have Romain Madeleine's group who is looking at sarcopenia and muscles in zebrafish. We have Eric Rogers' group who is looking at the aging in African turquoise killifish. So we have many different projects that have now come into action because of the availability of this particular microscope.
Question, anyone? Yeah, yeah.
Does microscope generate a lot of heat?
So just for our online audience, I'm going to repeat the question. And the question was, does the microscope generate a lot of heat?
Just in the media.
The microscope basically, it's the laser, that's where the heat comes from. But you also do not want samples to be heated up a lot. So the whole setup is kept in an air-conditioned room. So on a normal day, no, until something goes wrong.
If it's generating heat, you have a problem, like severe heat. Not really.
You want to add something, Fabian?
No, I mean, it's totally true. I mean, when I started doing microscopy, we had these giant lasers which would heat the entire room. Luckily, we don't need that. Very great in winter.
Yeah.
Any other questions? Yeah.
Did the system come with its own operating hardware, software, or did you have to write the software?
So the question is, did the system come with its own operating software? Do we have to write our own?
So they didn't have to write it, I wrote it.
So yes, it came with.
Yes, for me, though. I don't think.
You always get the German with your microscope.
Service operation.
Yeah, so when Fabian developed the microscope, obviously we also required a software to use this machine. And Fabian wrote the code that allows us to control various aspects of the microscope. And that code is what is open source, and you can just download and use it. And now after Fabian left Zurich, there is another person who is working on this same project to further improve it. And those things are also put on the open source platform where and it's distributed to the whole community.
Amazing.
So it's a developing system.
Yeah.
Yeah, which in the context of open source, it's a luckily we are in a very great situation that my previous lab, they were able to secure long-term funding, so on the order of 5 to 10 years, to continue development of this. Because one of the big challenges, of course, I'm working somewhere else in the meantime, and then of course somebody has to keep the code base updated. I should also say, actually, the software development is much more time consuming than the hardware development. So just it's like a factor of probably 5 to 6 longer than building the hardware, designing the hardware.
Maybe just so the audience understands, like you have your own research project. I mean, this is building microscopes. I mean, you're doing behavioral research in zebrafish on top of everything.
Yeah, that's what I'm doing now. But that mesoSPIM was basically the core of my PhD.
Okay.
Right. So that was like 3-4 years of almost full-time work.
That's amazing.
I think before we go to Dr. Haller, I just want to mention one more thing, and that is the amount of data that the microscope generates.
Yeah.
So on a traditional basis, when we do a sectioning, an image could be a couple of megabytes or up to gigabytes. But this microscope, obviously, we are taking so many images. The one thing more to take in consideration is that the speed of acquisition. The whole mouse brain that you saw there is acquired in less than 10 minutes. I mean, if we are to section an image in a traditional way, that would probably take, if not a month, at least a couple of weeks for sure. So, so this microscopy has allowed us to push boundary in many different ways, and we can acquire these images relatively quickly. Although the data is large, they usually run into gigabytes basically, but we are able to, now there are computational tools available that can handle those kind of data, which was probably not there 20 years ago. And so in order to have one technology evolve, it's important to keep in mind that many different players have to come together For example, we require expertise in chemistry to do the tissue clearing. We need an engineer who can build the machine. We need a software developer who can write the codes and solve all the bugs for operating the machine. We also need somebody who can do all the image processing like Frederick does for us. So it's not one person's job any longer. There are many people who come together to create such a beautiful technology.
Do you have a name for it?
mesoSPIM?
The microscope itself? You must have a personal name for it.
Okay.
That name? That's up to Marco.
Can I call it Marco? Marco one, Marco.
Marco used others.
Now, Prayag, I mean, we are all seriously impressed by the amount of data. Sounds, looks like the Mount Everest of data. We are impressed how many people you need there. Could you please tell us, since we know that even in high school, we needed somebody to tell us which things to turn to look through a normal microscope. The question is, what is the training time? How do you train? How do you, somebody comes now here from the audience, for instance, says, well, I'd like to do that. I want to do pictures like this. How does this work? Do you have an education process? Do you take their hands and then it takes 6 months, or how does it?
So just to summarize for the folks online, the question was, how long does it take to become efficient in utilizing this system? How long is the training process?
So this microscope is not a commercial microscope. So naturally, there are a lot of custom parts and there are a lot of custom tailoring done so that we can acquire this kind of images. So the training is not straightforward. In this particular case, Fabian taught everything to Marco and to some extent to me. I don't use much, it's mainly Marco. And then Marco transferred all the knowledge to Frederick. And well, that's how it is. There was a time I used to work on the bench. Now he's the workhorse. He does all the imaging. He takes care of the machine.
How many people, or let's have it the other way around, how many people at the moment at MDI or coming from the side are using the microscope and are able to use it?
So Marco transferred all the knowledge to Frederick, and at the moment, 2 or 3 people are currently fully equipped to use this machine. It's also important to keep in mind that the machine was only built last December, so we are only talking about last 8 months. Okay, so and I think that's a substantial progress. Basically, now from 1 person to 3 people who knows how to operate the machine, and there are more people who are interested, and slowly, steadily, Frederick is training them. So we reach there.
I would say it's 1 dedicated week. So you can, I mean, easy to learn, hard to master. But I mean, for everyday operation, I would say 1 dedicated week.
Yeah, it's not that difficult. I think the more difficult part is once you acquire all the images, how do you process those images? Because that's a lot of data. And just to give a perspective, I don't know about this machine, maybe Marco can correct me, but the previous light sheet where we used to acquire images, 1 mouse brain required roughly 25,000 images. Now we are talking about 25,000 images that needed to be stitched in 2 dimension and then into 3 dimension.
One second, with spinal cord, not
with
just
spinal cord.
not just the mouse brain.
Yes,
on its own. Then the mouse brain is depending on the resolution, but this what you see here is 1,500.
but even then it's still 1,000 of images basically. So how do you put them together? How do you make that they align with each other? It's like, you know, you have a Google map and you can keep zooming in to see what you want to see. So that's a lot of data processing and understanding and further rendering. For example, the kidney cases, as you saw, the extent of rendering that was required to get to the final product. So those are more challenging aspects than just learning how to use the microscope.
To give you just a perspective, like 20 kidneys I imaged in 2 days. The processing time will be probably 2 months.
Hmm.
So just the analyzation part. So we acquire data really at high speed. But one thing I also want to mention when you think about training people. So in this type of microscopes, you have a lot of degrees of freedom. When you think about a classic microscope, you have just a slide where you just put your sample in and there's not much to think about. But when you think about different samples, like let's say mouse brain or a zebrafish, or let's say different stages of zebrafish, it's always a question in which direction you actually put the entire sample. How do you place it? It's really not that trivial how you mount the sample. So this requires a bit of expertise. The machine to use or the mesoSPIM to use is not as difficult per se. I told Fabian yesterday, actually, for a custom built system, it's very straightforward and easy to use. And I was like, you did a really good job.
I agree.
Yeah, I mean,
And this is really a compliment.
yeah,
because-
custom systems can be sometimes crazy, like, and then it's full of bugs and things are just not working. So this thing was really neat, I would say.
That's great.
Yeah, and but what really takes a bit time and experience is then like just having an idea how you want to mount the sample. I give you an example with the mouse brain with the spinal cord. I use a dehydration-based method. So the spinal cord in a dehydration-based method would deform because it's kind of shrinking. Now, I was aware of this, so I kind of built a little cage around the spinal cords to keep it straight in the dehydration process. Because if you have a shrinked and deformed sample, kind of, then suddenly the amount of planes you have to image increases substantially. So even before you do the very first step, you have to kind of visualize and imagine how you want to actually acquire this type of images. Then you have like a 7 centimeter, which is 2 and something inch of mouse and spinal cord. So you need to think about what kind of chamber will I use? How do I put it in the chamber? So these are things which take a bit time, where you need to build up your experience.
If I may add some, there's something funny related to this, which is, it actually turns out, at least in my experience, that it's easier to train somebody who's never used a microscope before to use such a type of microscope than somebody who has lots of preconceptions about how professionally great microscopes should work. And one example is this microscope, you can rotate the samples. You can kind of pick the best view. In a normal microscope, you don't do that, right? You don't tilt your slide, right? And so that means it's people who are, let's say they have the prior experience, they just forget that there's the rotation, right? Because it's not there in a normal microscope. But here you can just rotate the sample to have the best view if necessary.
Or you actually need somebody who has a very vivid 3-dimensional or 4-dimensional imagination.
There is something funny related to that as well, which is, so my background is in neuroscience, right? And so I've seen a lot of people actually, so in neuroscience, you have certain, if people cut classical histological sections, you either cut them in certain planes, you cut them in this plane or you cut them in this plane. And that's the kind of also the view, you have like guidebooks, like an atlas, which tells you where you are in the brain. And of course, the images, they're also cut in these planes. And of course, if you can rotate your sample, you can have whatever plane you want, right? And suddenly you have people sitting in front of the microscope, and they've been studying the brain of a mouse for their whole life in a way, and they're totally lost in it, right? So they cannot, it's like this is their object of study, but they suddenly are not able to navigate. And if their background, let's say somebody has a neurosurgery background, but by training, they're really good at imagining this, like how it looks in 3D. So there is something, like there is a certain intuition you have to build when you suddenly are confronted with the full 3-dimensional extent of your sample.
Interesting.
I'm sorry, but I'm really fascinated by this because as a kidney doctor, the kidney is as complicated as the brain or even more. Although we have.
He's not.
our boss would have to say yes. Yes, of course.
What's really very important is that although we have done 100 years of research here, the three-dimensional structure of the kidney is still a mystery. When you ask any doctor on the planet or researcher in the kidney, how actually does the vasculature in the kidney work and how does it interact with the tubular system, they don't know. I mean, there are really only a handful of people who have a three-dimensional understanding of that. And a microscope like this then gives the opportunity to ask these questions.
Sure.
Yeah.
So I want to just talk a little bit about what you're excited about for the future with this microscope. So you're here to do an upgrade right now, right? In terms of increasing resolution. Are there other things that you can see kind of coming in the future? I want to know what I'm in for in terms of raising more money.
So of course, because I would say I'm the creator of this project, but I'm not the one, like, it's the tiny part of my current work for this project. But I can say from like where this project is going is like, so what we definitely see, it's also why the initial people who kind of approach me were like have a pathology background, is that some of the systems that are running, they see actually a lot of patient samples in terms of preclinical studies. That's mostly prostate biopsies for like patients with prostate cancer, and then sentinel lymph nodes for which are, so it's basically you know, a lymph node that's been removed in order to check whether there's you know, still some form of cancer somewhere in the body. And that stuff that's kind, but these are like you know, in a way very preclinical studies. Right? So this is not a system that's really intended for diagnosis on a daily basis. Right? It's intended to give researchers kind of the access to very, very high quality data. And so that is something that is happening on the like application side, which I think is most exciting. But it will be, you know, a long road before this is actually transferred into clinic. The other side is from a more technological point of view, you know, cameras are getting bigger, lenses are getting bigger. So, you know, and the amount of data, so, you know, there's people out there who now like have systems with 10 times the throughput of data. So it's definitely getting more challenging even handling and using bigger data. In terms of resolution, I would say that there's still a little bit one can push, but it's also not that easy to create a sample that's perfectly homogeneous, that you can have excellent resolution in the very center. That's something also where I feel like at some point when you know, still cutting the sample into pieces is not such a bad idea.
So you mentioned that this is just a small part of what you do, yet somehow you also had the time to have a new publication recently in Science where you talk about a new type of microscopy system, right? The Schmidt mirror system.
Schmidt object too.
Yeah.
That's yeah, I don't know. That's a kind of so I have a long-standing interest in astronomy, and I guess now people are wondering where this is going. For me, it actually was a very fun project because it turns out you can take an idea from astronomy, like how they build certain type of telescopes, and turn into a microscope. And the other weird thing is then that this type of microscope objective actually, if you look at it, it works very much like the eye of scallops. And so the very scallops that, you know, I guess here people eat on a like everyday basis, what you eat is a mussel, but actually the mantle of the scallop contains hundreds of eyes. If you've seen a live scallop, these are these beautiful blue little dots. Don't have it in here. And interestingly, these scallop eyes use a mirror, like a curved mirror, to focus the light on the photoreceptor. It's actually possible to build a microscope object. So it's, you know, sometimes you say, oh, astronomy has nothing to do with like microscopy, has nothing to do with scallops, but sometimes it comes together.
That's me.
It's not impossible.
I'm wondering, do we have any online questions?
Any online questions there, Sean?
Sure. Can you hear me?
Yes, go ahead, Dr. Warrior.
No. I'm interested in how large a specimen can you currently use in this instrumentation, and do you envision that this technology might eventually be adaptable the way an MRI scan, for example, is for the whole human body.
So the prototype I built is not telescope size. It's more like it's the size of a normal microscope objective. So this means like the mirror is roughly 3 centimeters in diameter. So the biggest samples you can look at is on the order of 6 by 6 millimeters. That prototype, if you want to look at bigger samples, you have to scale it. And so at some point it becomes really the size of a telescope.
I think he's also asking about mesoSPIM. What is the
General.
largest
So it's
sample?
in general, okay, but so that's more like a question for you then, I guess. How big can you make the cleared samples?
Yeah, so just recently a group published a decent resolution mouse,
Whole mouth.
a whole
Whole
mouse
mouse, adult mouse.
imaging. I mean, MRI. It's a bit difficult to make the human transparent. So, but one thing, I was talking about the kidneys from mouse, and actually the group we are working with first had a MRI look at the kidneys, and then they sacrificed the animal and gave me the samples, and I tissue cleared them, and then we compared the data we generated with their MRI data, and we had better resolution, and it was kind of nicer to see. And yeah, we can also specifically label certain structures, while in MRI you basically have just
So I think-
differences in
I think
magnetic
Jack
field.
has like 8 Tesla machine, and they tried MRI on kidney samples there.
I mean, it was okay, but okay.
And apparently the mesoSPIM images were far better compared to the MRI.
Yeah, and it's actually faster as well. So MRI, I mean, still it's a microscope, right? At one point, you will not call it a microscope anymore when you can look at the entire human. Okay. Yes.
Sally?
Sally?
If you're trying to extract data from a picture like this, and you want to know how many connections were done horizontally and how many connections were done tandemly or whatever, how much data, is there software for that? And do you have the computer power to do it?
I would say yes. And it's just a matter of resolution in the end. So with how much resolution we can image. Maybe if you look at this brain sample right now, can I have the laser pointer? So let's just focus on one of these neurons. In this case, the brain was recorded with lower resolution, so you don't see all the dendrites going away, and we are not speaking about little connections like spines on the dendrites. Now, yes, in theory, we can record things with higher resolution to, let's say, subcellular level. And I think if we can kind of stitch this type of samples, yes, it's very possible. It's not I mean, it is a challenge, but it's doable. Let's put it like this. It just needs time. It's being enhanced.
It strikes me that an important part of it is the decision of why you're using this machine as opposed to any number of the other machines, because there's a lot involved in the, once you get the data, you're going home and now you can't write the paper or anything. You still have a lot of work to do. So you might have been able to get all the information you needed in a simpler way.
Well, it depends on the question. So I was just explaining to someone other day, back in the days when somebody had to make a building, and if you go to an architect would take a big white sheet of paper and a pencil, and then he will sketch out everything. Now, in today's era, you have computer softwares like CAD, basically, where you can make a 3D map of architecture of the whole building and what will go where, how it will look. Both tells you the same thing, but the computation software gives you a very different perspective. When you see something in 3 dimension, first of all, it's mesmerizing to see such details, but at the same time, it gives you a perspective that you have never considered before. For example, if you're looking for the connections of neurons, no amount of 2-dimensional sectioning can do justice to the connections. You require 3-dimensional view to have an understanding.
The EM person would make the difference. So you can't do.
Are there any EM persons?
Yeah, that's what.
No, but this, goes into the same direction. Now, we are all impressed, and this is wonderful, and you just gave a nice example, what we can see now much better. And now the question is, when you analyze, for instance, here in greater detail, what are the questions? What will be the important questions you ask because you can see much better?
Exactly. So that's a good question. So Dr. Haller is asking why and how this 3-dimensional views is helping us to dissect out what we are looking. And to give you an idea, like if you label a neuron in central nervous system, and if you want to understand where the projections are ending, for example, in a regenerating limb, one can make a section of a spinal cord, one can make a section of a limb, and then you can guess that probably it's the same neuron who's connecting one end to the other, but it's still a guess. You know, when you see something in 3 dimensions running from one end to the other, you're not left with any doubts. Then you can. It's so confirming.
It's also connectome study. So how is the brain wired? If you think about how a neuron is actually projecting, it's never in a straight line. It goes like this, and then it branches off in many directions. With sections, it's just very, very difficult to reconstruct such a thing.
So this is great. I mean, we'll send somebody tomorrow because understanding why of the kidney, that's a question which urgently has to be answered.
We'll definitely take it out.
That's great. All right, so tune in next year. We'll be able to bring all these answers to you. Thank you guys so much, really, for your time this evening. And thanks to all of you for the great questions and for tuning in. Marco, can you advance that a couple of slides here?
Back,
Forward.
I understand also.
Sorry. There we go. So just a reminder that our next Science Cafe will be with Dr. Dane Disney and a colleague as we look at PFAS exposure. So we know that's a topic that interests lots of folks as we're learning more about the impact of these so-called forever chemicals in our water and agricultural systems. So tune in with us. Just a reminder, this is our last in-person cafe for the summer season. We'll revert back to our Zoom format for the winter. And I can't believe we're saying winter already. Okay, let's just talk about fall. But in any case, we encourage you to join us for that and look forward to another fun conversation with all of you. So thank you so much for being here. Take care. Thanks all of you.